Thank you, Beth. Thank you all for coming. Just let me know if you have any trouble hearing me. As Beth said, this is my thesis proposal, and I'm going to be talking about a study I'm interested in doing in Imatinib-resistant CML cases. And I'll talk a bit about the background of CML and resistance, about my aims, which will include driver gene identification and latent factor pathway analysis, and then some about the potential interpretation and significance of results that we might get out of this. So chronic myelogenous leukemia is a dual-phase leukemia, which is really famous for its clear genetic hallmark, the BCR-ABL translocation between chromosomes 9 and 22. This is the so-called Philadelphia chromosome. It was first characterized in 1960, and it was really the first example of a genetic hallmark of serious disease. And later work showed that this was the primary driver of CML in most cases. And this this translocation codes for a fusion gene, the BCR-ABL tyrosine kinase. And the way in which this affects the cell is by unlinking phosphorylation of downstream pathways which are involved in differentiation and proliferation of cells, and which is normally a very tightly regulated process. But this tyrosine kinase essentially spuriously phosphorylates these downstream products, leading to unregulated differentiation of myeloid cells. And this shows just a couple of the pathways that are activated that are known to play a role in the BCR-ABL pathway. The other reason that this is really famous is the development of imatinib mesylate, otherwise known as Gleevec. And this was done in the early 90s in collaboration between Brian Drucker's lab here and Novartis. And they, in this work, were able to successfully create a tyrosine kinase blocking drug. And this, again, was a really important result because it was the first example of a real rational targeted molecular medicine, and really kicked off sort of, in some ways, the idea of precision medicine, which has become a very dominant paradigm in modern genomics. And imatinib acts by blocking docking of ATP to the kinase domain of the BCR-ABL protein, and this prevents the addition of phosphates to tyrosine residues in the downstream targets of these pathways. Treatment with imatinib was really very successful, and this is just a figure showing some of the collected data from different clinical studies that looked at the effectiveness of treatment with imatinib. And you can see as much as 80% of cases showed complete cytogenic response after 18 months, which is a really excellent response to the drug. Unfortunately, this is primarily in chronic phase cases. There are two phases to the disease. The first one, the chronic phase, is characterized by a proliferation of myeloid progenitor cells, and this is largely driven by the BCR-ABL fusion gene. But the second step of the disease is progression into full-blown blast crisis, in which these progenitor cells differentiate and cause accumulation of granulocytes in the bloodstream and peripheral, or in the peripheral bloodstream and bone marrow, and really kicks off full-blown leukemia. And that process is a little less well understood at this point. And so the takeaway is just that while it was very successful, there are a significant number of cases which progress to the later stages of the disease, and imatinib has not been as successful in treatment of those, and their treatment options are fairly poor. Additionally, there are a number of cases, a pretty significant proportion, that acquire resistance to the drug over time. And because it's really a lifelong condition, it's something that is not cured per se, the proportion of the affected patients who acquire resistance over time increases and becomes fairly significant. In looking at the mechanism by which this resistance arises, a large proportion, roughly 50% or so of cases, exhibit some form of mutation within the BCR-ABL fusion gene. And this offers a really fairly clear, understandable mechanism by which the resistance arises. And this is just a ribbon structure of the BCR-ABL protein in green, and the imatinib molecule is outlined in blue. And you can see it acts by very precisely docking into the binding domain of the kinase and blocking the action of blocking the downstream phosphorylation that drives the disease. And most of the cases that exhibit point mutations have mutations within that region, and this causes a change in the steric conformation of the protein, thereby disrupting the action of the drug. And so this is fairly well understood. Unfortunately, though, roughly 50% of the cases of imatinib resistance in CML, the mechanism is not clear. They don't exhibit clear mutations to the actual BCR-ABL fusion gene. And so how this is occurring is still not well understood. And I'm going to just highlight a few things that have been implicated in the literature, just to illustrate the various ways in which this could be occurring. So this is just a basic schematic of the cell with the BCR-ABL gene and some of the downstream pathways that it affects. One of the ways that has been, one of the things that's been discussed is mutations which may be affecting the bioavailability of imatinib. And these are, there have been a few transporter genes that have been noted as being mutated at somewhat higher rates within patients who acquire imatinib resistance. And these may be affecting the rate at which imatinib is brought into the cell or cleared. And the important thing from this is just that this isn't really in any way perturbing the disease pathway itself. It's affecting something in the cell which is acting to block the action of the drug. Another example, a recent study here identified through an RNA screening experiment in cell lines certain genes which, when knocked down, led to resistance to imatinib. And what they found was that the knockdown of these genes led to activation of a CRAF pathway, and this acted on one of the downstream pathways known to be involved as a downstream product of the BCR-ABL gene. And so this acts sort of as a secondary entry point to that pathway, even though the action of the BCR-ABL fusion gene is still being blocked. A third one is just an observation that's been seen where a significant portion of patients who've progressed to blast crisis and have resistance to imatinib have shown a structural rearrangement where they lose one of the arms of chromosome 17, which contains the P53 gene. And it's thought that this might be acting as just a different sort of oncogenic trigger. And so in this way, a different pathway which could recapitulate the phenotype may be acting. And the point of talking about these is just that, I mean, what we're really interested in understanding is resistance. And so I think you have to broaden the perspective to the context of what's going on in the cell beyond just the disease pathway itself. And there are multiple ways in which this could be perturbed. Being able to effectively identify what that might be is a little challenging. I think a central question that comes up as a result of this is trying to understand if between resistant cases that exhibit mutations to the BCR-ABL gene and those that don't, whether or not they're activating the same pathways, and whether that means that there is some alternative mutation that's reactivating it, or it's something else entirely going on in the cell. So my specific aims, I'm going to be trying to do a few types of analyses to look at two separate things. One is identifying potential driver genes that are specific to this group of resistant CML cases. And the other is looking at the pathway signatures. And I'll be working with the Knight Informatics group on a project with Dr. Druker's lab where they've collected a large number of samples from patients who have acquired imatinib resistance, both those that have mutations within that gene and those that don't. This is just a workflow of what I'll be doing. We have both DNA-seq data in the form of whole exome sequencing and expression data in the form of RNA-seq. And we'll be doing genotyping and differential expression analysis. But all of those things I'll be doing some of it, but it will be in conjunction with the Knight the Knight group who have worked out very nice protocols for the best way to do this. And so that's something that will not be quite as interesting to my study. It's something I'll be doing with them, but the real meat of what I'm going to look at are identification of these driver genes using the results of our genotyping and some of the results of the expression data, and then pathway signatures, really trying to leverage this expression data. And I'll be using a couple of different algorithms, which I will go into in more detail. One of the interesting observations to come out of genomic studies of cancer in recent years is the fact that high-frequency mutations at the gene level are somewhat rare. More often, a large number of genes are disturbed at a lower frequency. But when evaluated as gene sets, oftentimes this problem can be overcome, and it may make more sense biologically to think of them as networks or groups of genes acting together within the disease. And so I'm going to be using two different techniques that attempt to characterize sets of genes to identify drivers of the disease, but in different ways. The first one is the Hot2 algorithm, and I'll talk about just roughly how this works. So starting with a list of variants that we've gotten from our genotyping, in mapping those variants to their respective genes, we would give each of the genes a score. And in this case, this would be based upon the frequency that the mutations are seen across the collection of patients. And then using this vector of inputs of mutated genes and then the frequency at which they're seen, we can overlay that onto protein interaction networks. And what this does is introduce sort of a topological element to it. I mean, it really brings in information, prior information that's known about how the protein products of these genes are functionally related. Once that's done, you're left with a network of genes, or a network where the nodes represent genes, some of which have scores based on their mutational frequency, and the edges represent interactions within this protein network. With that, the HotNet algorithm essentially acts by taking the heat or score of each of these genes, and then for each iteration sharing a percentage of it with all of its direct neighbors. And then as that repeats, you would take the heat going from one node out to its related partners would be spread out to secondary partners. And what this essentially does is allows us to identify groups of proteins or networks of genes that are functionally related, that have physical interaction in the cell, or putative interactions in the cell, but which are mutated across our samples in our study. And once you've done this, you can pull out significant networks, which may be representative of groups of genes that are acting together in the disease. Something I'm also interested in trying out, thinking about, is our various metrics for the gene scores. One other idea I have is to use differential expression between the two groups, those with the BCR-ABL independent phenotype and those with the dependent phenotype, and look at the differential expression for those mutated genes as a potential score. And I think that it would be interesting to compare the two, because it would really be looking at the mutations versus, in some ways, the expression. It could be different in a way that's meaningful. The second method that I'll be using for identification of driver genes is based on this idea of mutual exclusivity. And this is based on the idea of, as tumors evolve, new cells arise with new mutations. And if those mutations disrupt some biological function or pathway which leads to further differentiation, this can be thought of as conveying a selective advantage in a Darwinian sense and allows them to proliferate at a faster rate than other cell populations. And the idea of mutual exclusivity, given that, is that there are these sets of genes which exhibit a pattern where usually one and only one of the gene members is mutated in any given sample. And there have been a couple of studies that have shown that, well, within cancers, that these sets of genes are members of distinct disease pathways that are known to be part of a singular pathway structure. And so one of the hypotheses that's attempted to explain what this pattern might represent is that these might be identifying sets of genes which are acting that are members of the same pathway. And so essentially mutating one would cause this increase in fitness in the context of cancer, but past that point, mutation of another partner or downstream partner would have very little effect in increasing its proliferation. And so I think it's an interesting complement to the other method, in that this is identifying sets of genes that possibly represent a membership in the same pathway, as opposed to networks of genes which are potentially acting in the context of the disease but are not pathway members. And this is just a schematic showing the basic idea. If you have a number of samples from different patients and you look at the mutation of all your various genes, these methods identify sets of genes marked in red here, where, if evaluated on their own, one single mutation is found for each gene, or for just one member of each gene set across the samples. And that's the basic idea. This is a really simple toy example, obviously. And there are a couple of algorithms out there that have attempted to identify gene sets this way. I decided to use one called Multidendrix, and there are a few reasons. It's not rigorously tied to any sort of prior information, so it's identifying them in a truly de novo fashion. And it's able to identify multiple sets of genes exhibiting this pattern simultaneously. So for AIM2, I'm really interested in doing something that's able to give a more of a context of the expression of these genes rather than looking at just the mutational landscape. I think the driver gene sets are very useful, but it's a fairly flat picture of what's going on in the disease state. I think having something that really leverages expression data to give a fuller picture of pathway activation is really useful. And many of the more commonly used approaches for pathway analysis, like gene set enrichment analysis, look at some measure of expression like differential expression, and then test for enrichment of those differentially expressed genes within certain predefined pathways. And there are some downsides to that, though it's a methodology that has been well validated at this point. One is that they generally evaluate pathways individually, which can present a skewed picture of pathway activation because of crosstalk between pathways. And I'll talk a little more about that in a minute. But the main thing is that they're pretty tightly constrained by our prior knowledge about pathway membership. And latent factor analysis is another technique that has been used in a few papers for this sort of pathway analysis that offers a less constrained way of looking at it. And I'm interested in trying this out. In the latent factor model, we attempt to explain the correlation between all the known variables, which would be represented by the genes we're studying in these expression data sets, in terms of a smaller number of unknown variables, or latent factors, which could represent compound groups of genes. And in that way, it acts sort of as a gene, or a data reduction technique. And I'll just make a few points about how this works. And basically, what you have as a result of your experiment is this matrix of expression values, shown by the response matrix here, where you have a number of samples and you have some sort of expression output for each of the genes. And the latent factor analysis models this as a linear combination or a linear product of the pathway signatures for those genes and the sort of true ground state of pathway activation in your sample, which is really what we're interested in, and we don't know. In doing this, it's able to extrapolate or pull out the pathway activation that's causing the pattern of expression that we're seeing. And factor analysis has its own set of issues. Interpretability of the latent factors can be difficult, so you can sometimes combine a number of genes that don't have a real obvious common functional context. Pathway crosstalk is a real issue, and this is a product of the fact that pathways are really oftentimes hierarchical, and one pathway can be a subset of a larger pathway. Many pathways share gene members, so if you're doing a pathway analysis and you really want to understand what is driving your disease, you may have one or two pathways which are driving it, but are also affecting other pathways with which they share genes. And so in terms of interpreting what you get out of it, this is an issue you have to take into account. And then just the type of data that you can use as inputs. Some of the methods that are out there, there's not too many, but a few of them use just simple gene set lists, as you get from, say, a KEGG pathway database, or they might use actual differential expression or magnitude of expression values, all of which give a little more detailed view of what's going on. And I'm proposing to use a fairly recent model called ASSIGN from the Johnson Lab in Boston University. And the important thing here is just that it's a fairly flexible version of this latent factor analysis framework that allows, or that accounts for a few of these issues that I brought up. And it allows for the evaluation of multiple pathways simultaneously, and they've shown that it does a good job of avoiding pathway crosstalk. And it also allows for a variety of different inputs as pathway signatures, either gene sets or actual magnitude expression values. And I think comparing the results of these two aims could be potentially pretty interesting. I think that looking at the potential driver genes that are identified using these methods, and looking at looking at their membership in pathways that are identified between the two groups of CML patients could give us some clue as to what's going on if they're present in pathways which are not differentially expressed between the two groups. That could be indicative of reactivation of the same pathways seen in normal BCR-ABL driven CML. I think if they're enriched within pathways that are unique to that are differentially expressed between the two groups and unique to the BCR-ABL independent group, that could indicate a different mechanism is being used. And I think it could be interesting. I think it would be interesting and add some context to be able to understand what this means. And another thing is that I'll be focusing most of my analysis on chronic phase patients because I think it makes the most sense clinically in that blocking the transition from chronic phase to the blast crisis phase is really how Imatinib is able to successfully treat the disease. And I think something that is identified in that phase is more likely to be meaningful in terms of secondary treatments that might have some effect on preventing resistance in these cases. But we have a handful of samples that are from newly diagnosed patients who are on Imatinib treatment but have not acquired resistance. And so I think looking at some of the driver genes and pathway signatures that we find in those samples to see if they're present early on and whether they potentially could be predictive in any sense is something that could be interesting as well. And then as far as the significance, I think it would be very useful in understanding potential targets for secondary therapies. I think it also could, this resistance could be acting through similar mechanisms as what is seen in normal CML in transitioning from the chronic to blast phase. And I think it could be interesting in the context of that. And I think it also could be interesting in understanding the mechanism of atypical CML, which doesn't exhibit a canonical BCR-ABL fusion gene. And in conclusion, I just want to thank my committee, my advisor Beth, and Christina and Shannon for all your valuable feedback, and my family, who've put up with a lot of stress and neglect over the last 2 years while I've been in school. So yeah, thank you.
So when you, for the first HotNet method, you've identified genes and put in, can you describe the hot-cold scale that you plan to apply to those genes and the criteria that you want to identify as being hot or cold?
Yeah, well, I mean, I think, as I said, the most basic metric for that's used in this is just the frequency that they're seeing across the samples. And so a larger value or hotter score for each gene would be for those, obviously, that have a high frequency of mutation.
Do you think that you'd actually see would those be the ones that you would believe would be more, you went through the sort of the long tail of cancer, and that most of them are not frequently recurrent mutations? Do you think that that the hot ones should be the most highly recurrent necessarily, or?
I think so. I mean, it's still, I think, operating under the idea that the mutational frequency, even though they're not seen at high frequency, that is indicative of their significance in the disease. I think the thing that really lets you get around it is the structure of the protein interaction network. And that's what's really identifying the networks of genes. The scores do sort of reinforce that and allow you, I think, to pick out more easily important networks and reinforce and increase the, I guess, the heat signature of those networks that have both a lot of connections and a significant amount of mutation within their group. But I think the thing that's really identifying them is the protein interaction structure. I don't know if that answers your question.
If I could just,
Nathan?
Yeah, I just would like to add to the discussion about that, because I know we talk about this a lot. The difference between an individual personalized medicine approach, for which we've developed algorithms to look at an individual person, but also in a population context across samples. So we look for, yes, mutations that are in more than one, but we don't want to get to the point of polymorphisms in the population, so we don't want to get too high. So there's that balance between more than a few and not too many. But also, one of the things that has to be carefully thought about in this whole project and projects like this is the inputs of the variants that we actually input in and how we screen those out from all the WES. So there are several ways that we do that, but that's one really important aspect of determining which mutations we would actually even want to use as inputs, which we work with the lab on that.
Nathan?
So if I'm understanding right, you're using the mutation data with HotNet to kind of find some genes there, targets there, and then expression data with the more matrix factorization type idea. And I'm wondering about using the heat from the mutation data, but instead of protein-protein interaction networks, applying that to your latent pathways that you discover from the other one. If you could combine those two together.
I mean, I thought about that early on when I was sort of working on my thesis proposal. I think you have to be careful to not sort of double back on yourself, and in identifying groups of genes that are mutated with a certain frequency, and then these like driver genes, and then using those as your pathway signatures. You're drawing from the same data set that you're analyzing. And I think in the factor analysis, having predefined pathway signatures that are outside of your data set is an important aspect of it. And so I think I was a little worried about the possibility of it being a bit circular, but I'm curious to think about it a little bit more.
Yeah, I see what you mean. But I was thinking more, I mean, because the mutation and the expression data are somewhat separate. I mean, they're different types of data, at least.
Yeah, they're different types of data. I mean, they're from the same.
Other than just looking at basic overlap of what you find on hits.
Yeah.
I mean, that's a bigger problem in general, combining different types of data.
Yeah. Yeah, it's something I'd be interested in investigating. I mean, I'm really interested in methods that can, in some way, integrate these different types of data in a meaningful way. And it would be really nice if there was a way to do that. But I'm not exactly sure at this point how that would happen. We'll see. Ted?
Do you have any thoughts about how you're going to present your final results to researchers?
I mean, I think with this sort of a study, there's no way getting around trying to work with the lab after identifying some of these in order to see if there's a way we can validate or do some more experimental work to look at whether this is something real. I mean, this is I think the downside to some of these, I guess, machine learning approaches in some ways, is that it's hard to know whether what you're seeing is real at all times. And we do have known BCR-ABL driven pathways that could act as some sort of validation to see whether, like, what I'm getting out of this makes any sense, see if some of those things are included. But I think that, it would mean, coming up with like a list of potential things and seeing how that agrees with some of the other literature that's out there and talking with the lab about possibly finding a way to test that.
Do you think that there's a finite number of pathways that would be activated in the BCR-ABL independent mechanism? I'm just thinking that, if it's still CML, there's going to be CML pathways that are activated to maintain that phenotype. It's not transitioning to AML or transitioning to some other type of disease. So you're really looking at some finite number of pathways that are going to still be activated to produce that type of disease. So is there a way that you can kind of start to cull that down a little bit so that you hone in on some of those pathways?
Yeah, I'm not entirely sure, to be honest. I mean, that's a really good point. I mean, I think that in thinking about this, I'm not sure whether, I mean, my understanding of cancer biology, admittedly, is in an early stage, so I guess I don't have a lot of experience to draw on with this, but I did spend some time thinking about whether, it makes sense that you would see a number of different pathways causing this same phenotype, or if it would be likely to be just a couple, and I'm not sure exactly how to account for that.
Part of that is taken into consideration again in the choice of the input pathways for the assign algorithm. So that's another, inputs are always really critical.
Yeah, I mean, I think for the pathway signature inputs, I'm hoping to use a couple of different pathway signature data sets that kind of are at different levels of contextual application. Some more general ones, and then there are some data sets that are more specific to cancer and more specific potentially to leukemias, and see if, I think, in looking at multiple data sets that are sort of narrowing down, maybe that would be a way to filter some of that out, but I'm not sure.
Do you know, again, based on the way your aims are set up there, you're profiling driver genes being mutated as per the detection of variants via whole exome sequencing. In the event that, for example, there's a newly acquired cryptic translocation or something, whatever, new fusion in a resistant sample that is the principal responsible reason for the resistance, and thus something that wouldn't be detected in the setting of whole exome sequencing, but in the setting of RNA-seq would. Is there a way to incorporate any of potentially interesting driver genes or fusions from the RNA-seq data into the level of the analysis in the driver gene network?
So, I think both of the driver gene methods allow for input of multiple types of mutational data. I think my plan is to focus on the single nucleotide variants because we have a more robust protocol for determining those in this sort of a case where we don't have normal controls, because we have to do a little more to try and pick out what we would consider actual somatic variants. And there are methods for identifying, like copy number changes or structural rearrangements, but without matched data, they're a little less certain. And so, I mean, those things can definitely be incorporated into both of those methods, I think, but it would be more an issue of having confidence in those calls. Thank you.